Hi everyone, I'm Derek Stewart. I'm the Director of the Advanced Plant Growth Centre, which is a regional development project at the James Hutton Institute. It's a £27 million project looking essentially at pre and post-harvest technologies for things like controlled environment agriculture, vertical farming. It's also including large investments in plant phenotyping, post-harvest storage, things like that. So as part of the programme of development of this new centre, we have set up a seminar series, and the first one is today. And in these, we want to talk about advances in all of these areas, and by and large across plant science per se. So to kick it off, we have today, probably known to many of you, Rob Edwards, who's head of department at the School of Agriculture at the University of Newcastle. Rob's background is a degree, a BSc in Science Biochemistry in Bath, then he went on to Environmental Toxicology, quite a trendy area at the moment, it's got to be said, in the University of London. And then from there, some interesting jumps to head of department at the University of Durham, after working in industry in the Nobel Foundation. And then he became the Chief Scientist at FERA, the Food and Environmental Research Agency. And then from there, stop at University of York before becoming head of School of Agriculture, Food and Rural Development at the University of Newcastle. One of the few schools of agriculture left in the UK, it has to be said. So flying the flag. If you look at Rob's publications on Google Scholar, you can see it's the genome, as he calls it, is his focus of research. And so that's the comprehensive chemical detoxification systems that plants have, and looking to see how we can exploit that to basically develop the next generation of control systems for things like he's talking about today, wild grasses. So, Rob, if you'd like to kick off, the floor is yours.
Okay, thanks very much, Derek, and thanks very much for the invitation from the centre and from James Hutton. So what I'm going to talk about today is some work that we've been doing really since about 2017 on herbicide resistance in wild grasses. But to add a bit of topicality to it, what I'm going to be talking about today is some technology that we've developed to help diagnose herbicide resistance in wild grasses that shows some uncanny correlations to the way in which we've had to deal with COVID-19 over the last couple of years. And in fact, what we're dealing with here in terms of herbicide resistance in wild grasses in arable agriculture is a pandemic of its own type, which is seriously impacting on cereal production in the UK and further afield. So I'm just going to break the talk down into, first of all, why do we need herbicides? And I'll give a bit of the usual background blurb on global food security, but I think it's important because at the moment, with this great emphasis on net zero, we have lost a bit of perspective on why we actually farm the land in the first place, which is to grow food. And I'm just going to re-emphasize the fact that we do need that in the years ahead. Then I'm going to talk about the challenge of controlling wild grasses in cereal crops and why that's really important to get a handle on. And then when you use chemicals to do that, what happens when it all goes horribly wrong? And that's when I'm going to start talking about the herbicide resistance, especially in our favoured weed at the moment here in England, which is blackgrass. I'm then going to talk about how, just as we've done with COVID-19, by understanding what was the basis of the infection and the virus and how it actually impacts on us health-wise, we've done exactly the same kind of things around understanding the molecular basis of herbicide resistance in blackgrass. And in doing so, we've been able to develop practical tools that help us to counteract it going forward, including these point-of-care resistance diagnostics. And then at the very end, I'm just going to talk a little bit about how I can see diagnostics working in terms of herbicide resistance, and also a little bit more widely on how we can counteract the problem going forward. So this is just a slide. I'm sure you've seen various types of similarities in the numbers, but it really at the moment we've got this whole thing of farming for the environment and net zero, which are fantastic, laudable goals, and yes, very important. But we do have to feed a lot of people in the next few years, and it's been calculated here that we're going to have to produce as much food in the next 40 years as we had in the last 10,000 combined. And when you think of the size of that challenge and the fact that we're going to have to do it in a more sustainable and productive manner, it's a massive, massive challenge for us all as agricultural scientists. And I guess that's one of the reasons why your Advanced Plant Centre is going to be so important going forward. It's not just the fact that we're going to have to produce more food, we're going to have to produce food perhaps which has a different nutritional balance to it. So, it's just flagging up the fact here, there are as many people who are overweight in the world as there are undernourished. And so it's not just producing more of what we've got, it's actually producing food of higher quality as well as improving yield. Now, set against that, you'd expect agriculture to be rising to the challenge at a rate of knots. But actually, when you start to look at the numbers, you can see that something's not kind of quite right here. For many of the world's most important crops, the increase in productivity, and this is just yield, is pretty much either static. In some instances, it's gone up very slightly year on year. In quite a few cases, it's actually going down. So there's something kind of not quite right because, there's a lot of technology going into improving these crops, both genetically and through the agronomy and all of the kind of agrochemistry that sits around them. And yet we're not seeing those big returns in terms of increased productivity and yield. So we can see that really clearly here in the UK with winter wheat production. Winter wheat productivity in terms of tons per hectare kind of peaked out at about 8 tons per hectare in around 1995, and it's kind of stuck there ever since. And that's in the face of things where we know from the genetic improvements that we're putting into wheat, we would be expecting something around like 1.7% improvements in yield year on year. And as the years have passed by, that has created quite a large gap between what we're actually producing now and what we should be able to produce in terms of theoretical yield. It's something called the yield gap. So something's holding us back here. And there are many reasons for it, actually, and it's a talk in its own right. But one of the things that has hindered us, as it does hinder all of global crop production, is the issue of crop protection. So this is a histogram just showing the effects of the value of crop protection. So this is protecting crops against insects, disease, and competition from weeds through the conventional application of technologies, as we've shown here in the red bar. And then there's the bit at the top, which is a really interesting bit, which is the green space on the histogram, which is the bit of production that we are losing in these different crops because we're unable to effectively protect them against those different challenges. But, it is in its own way a great success story that we are able to provide so much more of the world's food than we would if we didn't have these sorts of technologies available to us. But importantly, if we can just close that loss that we're seeing in that green segment of the histogram, we would be able to feed many more millions of people without having to put any more land into production or any greater inputs into agriculture. So my own area of interest over the last 15 to 20 years has been on weed control. And, very often we don't actually take weeds very seriously. We think of them as being a slight irritation. In some cases, quite like you're seeing weed, but if effectively they are the greatest or insect predation, and that would vary from country to country. But here in the UK, the greatest constraint that we have on our production in a normal year is on competition from weeds, and in particular, in the case of winter wheat, from wild grasses. Now, weeds come in lots of different flavors. There's over 30,000 species of plants have been described variously in the literature as weeds, and that's normally in specific cropping systems. But if you really look at the real bad guys, the ones that keep reoccurring, you're talking about 100-odd different species, which tend to be really problematic weeds in the major crops around the world, and they keep consistently coming up time and time again. And it's a continuous battle when you're dealing with weeds because they shed so many seeds. The seeds build up a seed bank within the ground, and that can last for many years. In fact, for many generations, that can become a problem. And so it's been calculated, for example, that in the US, a typical acre of farmland will have anything between 50 to 300 million weed seeds just waiting to pop up out of the ground in the future. So it's something you have to keep on top on year, season on season. So at the moment, our major technology available to us, particularly in broadacre crops to control weeds, is through the use of chemical herbicides. And so the question these days is, chemicals have become increasingly unpopular in terms of why we're using them in the environment. So why do we use herbicides? Well, from a farmer's perspective, there is a very strong cost-benefit analysis that they can perform on the use of herbicides. They give them a good return on their investment in terms of returns on yield for the costs of the chemicals that they have to spray. They are also able to scale up the application of these compounds to very large fields, so they're applicable to hundreds of hectares of land. And in terms of practicality in arable crops, where you've got something like wheat or barley, where you've got plants that are planted pretty close to one another, actually the alternatives for utilizing chemical methods for control are relatively restricted. You're going to cause quite a lot of crop damage utilizing a lot of manual weeding, for example. Also, and it's quite often forgotten, herbicides can actually reduce the carbon footprint of agriculture. They allow farmers to move to zero tillage agriculture because they can keep down the number of weed seeds that fall onto the ground. That has advantages not only in terms of reducing diesel use in terms of ploughing, but it also allows, of course, habitat for animals to have and other organisms to have over winter. And of course, if we don't have chemical control, we need to go back to something that we used to do in times before, which is manual weeding, described as the greatest single endeavor of man across the planet. And of course, in many continents and countries, it isn't the men that do the weeding, it's actually the women. And it's back-breaking work. It's not the sort of thing that most of us would want to give a lot of our time to in terms of a means of controlling weeds on our major crops. So in terms of herbicides, a little bit about their terminology. They come in two major different flavors. There's total herbicides and there's selective herbicides. Total herbicides are compounds like Roundup or glyphosate, and they will kill both crops and weeds with equal efficiency. Then there's some rather more interesting, to my mind anyway, herbicides which are used selectively, and these will selectively kill weeds, but they won't kill the crop. And so these are very useful when you're using them in post-emergence applications when the crops and weeds are up growing together. And that brings us on to the second bit of terminology. You can apply herbicides either before the plant emerges, the crops emerge from the ground, which are pre-emergence compounds typically applied onto the soil, or you, rather more interestingly from our perspective, use post-emergence when you're applying them at various points of the growing cycle of crops to control the competing weeds growing alongside your crop species. The other thing about herbicides is, although there are many different potential ways in which we could inhibit and kill plants by knocking out different metabolic pathways, in fact, there are many hundreds if you do the kind of metabolic analyses, actually the numbers of those modes of action which have ever been usefully exploited are down to probably about a couple of dozen. So it means that we're very dependent on a relatively small numbers of ways of inhibiting plant growth in order to be able to control weeds. So we've got a restricted number of modes of action. And typically that means that we're hitting the plants time and time again with compounds that are acting on the same modes of action. And you can, for those of you who have a biological background, you can start to see that this may not be such a great thing in terms of selection and evolution. Importantly, no new mode of action has been commercialized from a herbicide perspective since the early 1980s. The reasons for that are many, potentially linked in some instances to a shift in the business of agrochemical companies away from chemical discovery into biotechnology. But still, we haven't seen that recurrence of discovery in anything like the way that we were seeing up until the 1980s. And that was important because the farming community became very used to new modes of action coming through. And that meant they had a series of sort of new silver bullets that they could apply in terms of controlling weeds in different ways year on year. It typically takes about 10 years to discover and develop a new herbicide from the day you synthesize it to the day you sell it as a product. And it can cost several hundred millions of pounds to put it through that discovery cycle. But having said all those things, there have been some massive advances in herbicide technology since they were first developed and used in the 1950s. So typically in the early 1960s, we'd be using compounds at several kilograms per hectare to control weeds. And now we have compounds that work down at grams per hectare. So, 1,000-fold increases in the efficacy of these compounds. So 1,000 times less of those chemicals going into the environment than they would have done 60 years ago. Here in the UK, we're actually very major users of herbicides, in fact, major users of all sorts of agrochemicals. Typically, no great surprises as to where they're used the most. They're used most in the intensive arable areas, typically in the eastern southern part of England. And also illustrative in this particular slide is the fact that we're applying these compounds several times through the growing cycle, and we're growing them and we're using them alongside typically fungicides as well. So these are compounds which are used not just once during the growing cycle of a crop. They can be used on many different occasions to control weeds as the crops develop. So on to one of the great challenges of agronomy and arable agriculture. Very typically, when we are growing crops like wheat and barley, we need to suppress the growth of competing weeds. The weeds will compete with them. And as I'll show in just a few slides, they can be very effective at reducing yield by just robbing the nutrients and in some instances competing for sunlight. And the most problematic weeds that we have in cereal crops are grass weeds. And it doesn't take an enormous sort of step of understanding here to realize that what we're trying to do is control a non-domesticated grass in a domesticated grass, which is your cereal plant. So effectively, physiologically, the weed and the crop are very similar to one another. The only thing is one of them is the plant that you want, and the other one is the one you don't want to allow to get established. So there's real conserved physiology between the crop and the weed, which means there are relatively few ways that you can exploit to control the weed in terms of hitting it with a mode of action that's not going to have an impact on the crop. In fact, the ways in which we do utilize these compounds selectively is relying on something that we don't entirely understand why it happens, but crop plants are able to typically metabolize and detoxify the herbicides many times faster than we see in the non-domesticated grass. So when you spray your herbicide on your mixture of weed and crop, the crop is able to metabolize the herbicide really quickly and it evades toxicity. The weed metabolizes the herbicide slowly and is actually affected by it. Very typically now, in fact, in terms of controlling wild grasses in cereal crops, we're very dependent on a very small numbers of modes of action, typically those which impact on fatty acid biosynthesis and branch chain amino acid biosynthesis. If selectivity, so if we lose that ability to differentially metabolize the herbicide between crop and weed, we will lose selectivity. So at that point, once you've lost the differences in rates of detoxification between crop and weed, the two plants will become equally sensitive to the herbicide. And this is how effectively a very simple schematic of just showing how plants undertake those detoxification reactions. In fact, the reactions that are being carried out here are almost identical to the ones that happen in our own bodies, most typically in the liver when we ingest foreign compounds like drugs. It's what we call the xenome, and it's a series of enzymes which introduce functionality into the drug, or in this case herbicide, that then allow you to bolt on large polar molecules like sugars and peptides that take away all the toxicity of the molecule. And once you've taken away all of that toxicity, what you will do if you're a plant is put those compounds into the large vesicular body that's present in all cells called the vacuole. And at that point, the job is done. You've detoxified the herbicide. And if you do it quickly enough, you can evade any toxicity as a consequence of the herbicide by accumulating in the cell. And this is just a very simple schematic showing the detoxification of a herbicide called rimsulfuron. And in this instance, what we're seeing here is on that ring, that aromatic, the cyclic ring on the left-hand side, the pyrimidine ring, you see a hydroxylation reaction occurring. And it's the introduction of that OH, simple OH group into the molecule, which takes away all of its toxicities, because now the herbicide is unable to bind to its mode of action site. And then typically the job will be then completed by a second enzyme, which will bolt a sugar onto it. And at that point, the compound is ready for vacuolar storage. And these reactions can occur incredibly quickly in your crop plants, but tend to occur very slowly in weeds. Now, in terms of our own kind of favoured weed of interest at the moment is blackgrass, Alopecuous myceroides. This is not a native grass of UK or indeed Northern Europe. It was brought here by the Bronze Age farmers who brought wheat and barley with them as well. And it's a grass that probably arose originally in the Fertile Crescent around kind of Syria in the Middle East. It's a plant that's been in the UK for many hundreds of years without causing that much bother. It's been described for a long time. It was never really an agronomic problem. It's an outcrossing, obligately outcrossing plant, so it acquires genetic diversity very rapidly. And importantly, it's got a growth pattern that is identical to that of winter wheat. So it'll start to germinate in the autumn, in September, October time, and then it'll overwinter and then recommence growth in the springtime. So it can actually kind of follow the growth pattern of the plant, which has now become a really serious weed too, which is winter wheat. This is a plant which was first started to become a bit of a problem in terms of weed control in the 1980s. And it went from being a relatively minor irritation in the early 80s to it was the number 9 major agronomy control issue in the UK in 1989. But by 2009 and onwards, it's been one of the single biggest constraints and problems in terms of weed control in winter wheat. No. And this is why it's a problem, because you don't need very many of these plants per square meter in a wheat field to give yourself a major problem with respect to yield. So just 12 plants per square meter can typically give you anything between 5 to 15% losses in yield. So this is a weed that once it becomes established in your wheat field can cause very significant losses of quality, both quantity of wheat and also potentially of quality. It's a species that can actually harbour ergot, so it can also introduce quite a lot of unpleasant toxins into the food chain as well. So where do we get to with respect to having this weed that was not much of a problem in the 1980s to becoming a massive issue now? And it really arose from the makings of herbicide resistance. And we can examine those rationale reasons as to why we've got this problem, in as much that these are the major things that have driven it. First of all, limited rotations and cultural practice. We've become very dependent on growing, particularly in broadacre fields, a relatively small number of crops, and we grow them year in, year out. Oilseed rape, barley, wheat, and again. We're highly dependent on controlling the weeds that afflict those crops with chemicals, so we don't tend to use other control measures. And over the last 30 years, we've seen a large-scale reduction in the numbers and variety of herbicides that are available for us to use, largely as from the constraints of the environmental and toxicological concerns that are around some of those chemicals driven through the European Union's precautionary principles. So very few new modes of action have been developed, so we haven't got new chemistries coming through, and you've got this genetically diverse set of weeds. So what we've been doing is we've been doing the same old thing year in, year out, applying the same chemistries again and again on a population of weeds which is highly genetically diverse, and you don't need to be Mendel to work out where this is going. What we've done through this massive selection experiment is start to select weeds which are resistant to herbicides. And this is what it looks like when it all goes horribly wrong. This is a field of winter wheat growing just outside of Oxford. It was filmed 3 or 4 years ago. And this field, as you can see, there's something badly wrong with the wheat. It's full of this dark grass, which is blackgrass. And this whole field would have been lost in terms of its viability as a crop. The whole lot would have been put into an anaerobic digester before the weed seed set, and the whole crop would have been lost. This is not an uncommon scene now in some parts of southern England. You do get these mass infestations, which can cause catastrophic losses in yield. So just to recap here, this is a weed problem. It's only emerged in the 1980s. Until now, it's become one of the biggest constraints on winter wheat production in the UK. It affects well over 1.2 million hectares of our best arable land. This is actually sliders are now 3 or 4 years old. We would see the black grass range now moving well into Yorkshire. In fact, it's not that very much far south of us here, up in Northumbria. And I'll go into the kind of numbers in a moment, but it costs us a lot of lost yield, and it costs us a lot of money in terms of our farming community. So typically it's up. Some years it's as high as 0.5 billion. In fact, the numbers I've got in just a moment will say something slightly less than that. So the question is, it's a big problem which we really haven't allowed ourselves to get on top of, and now it's come back to haunt us. This is just a breakdown of what those losses mean in a typical year. And in terms of the winter wheat yield losses, you can see that by far and away the parts of the country that are hurting the worst from losing that yield are down in the eastern and southern parts of England. And in terms of what it means is that typically at the moment we're losing about 0.8 million tonnes of winter wheat productivity, worth around about 0.4 billion pounds a year. If we do the modelling and we could show how bad it could get if we continue on the way that we are at the moment, that could rise to losses of 3.4 million tonnes a year and over £1 billion worth of lost production. So this is a really, really big issue for UK arable farmers. And you in Scotland are actually kind of quite fortunate in many ways that the blackgrass is not such an issue for you, but certainly in the southern parts of the UK, this is really hurting a lot of our farmers. Now, in terms of herbicide resistance, we can see that herbicide resistance is a big problem in wheat in terms of the numbers of different weeds that it's been reported to have actually evolved herbicide resistance in this particular crop. But you can see it's not unique to wheat. We see it with all of the major cereals, and in fact, all of the big arable crops. They all have associated herbicide resistance problems, with multiple weed species now acquiring resistance to herbicides. And it's a global problem. So we're not just seeing it here in the UK. We're not just seeing it in Northern Europe. It's a massive problem across North America, in Australia, but it's now extending well into southern South America and also into Asia. So wherever herbicides are used, herbicide resistance tends to follow, and it tends to follow not that many years after you start to introduce new herbicide chemistries. Now, there are two different types of herbicide resistance, which I'm just going to describe here. Firstly, we see something called Targetsite resistance. Targetsite resistance is caused when you have mutations in the DNA of protein, which encode proteins which are targeted by the herbicide. So mutations which cause changes in the DNA sequence can cause changes in the protein sequence. Changes in the protein sequence alter the shape of the protein. The protein no longer is able to bind to the herbicide. Herbicide no longer inhibits that protein, and therefore you've evolved what's known as Targetsite resistance. This is a highly effective way of achieving resistance to a specific chemical. The only downside in terms of it as being a resistance mechanism from the point of view of the weed is it can be overcome by rotating modes of action. So if you now go to a different mode of action, you can still kill that weed because you're avoiding that particular target site which has undergone those mutations. But this is now very common as a primary mechanism for the evolution of herbicide resistance in wild grasses. And then the other type of resistance that we see is non-Targetsite resistance. Non-Targetsite resistance is, as its name describes, everything that isn't Targetsite resistance. Its primary mechanism actually is enhanced metabolism. So if you remember just what I told you a moment ago about metabolism being the basis of herbicide selectivity, if you get enhanced metabolism of herbicides in wild grasses, you start to see them becoming as tolerant to the herbicide as the crop plant. So you're going to need as much of the herbicide now to kill the weed as you will to kill the crop. So you've lost selectivity, and effectively you've lost the use of that herbicide. Non-Targetsite resistance behaves very differently from Targetsite resistance. It's quite complicated. So it involves more than a single gene. It can involve dozens of genes. It's also what we call a quantitative trait. So it can range everything from very modest resistance up to very severe resistance. And the other important thing about it is you can't get around it by rotating modes of action. In fact, quite the contrary, as I'll come back to towards the end of the talk. So the real worry here with non-target-site resistance is you could develop a new herbicide with a new mode of action. You could spray it on one of these non-target-site resistant grass weeds, and that grass weed may be already primed to be resistant to that new herbicide because it will just chew it up and metabolize it in much the same way as it would do with any other chemical it comes across. So this isn't just a resistance problem for herbicides we have now. This is potentially a herbicide resistance nightmare for compounds yet to be discovered. So when you've got these big problems, you obviously can't take them on as a single research group. And so in 2014, we put together a consortium with the University of Sheffield, Rothamsted, University of York, and the Institute of Zoology, as it was. And we pulled together a grant where we wanted to look at the fundamentals of the evolution, biochemistry, and ecology of herbicide resistance, but not just work in our academic ivory towers, but actually integrate the knowledge that we were generating throughout that project with the farming and agronomy sectors. So what we didn't want to do is do 4 years worth of work and then come and tell the sector afterwards what we'd done. We wanted to work with everybody to explain what we were doing and get some guidelines as to where they saw the greatest innovation, the value of the innovation going forward. So we were the leads on work package 1, which is understanding molecular mechanisms of resistance. You know, we're biochemists. We like to understand how things work at the molecular and cellular level. And all of our work packages were involved in generating fundamental knowledge. But importantly, each of our work packages was also primed to have a practical output. And the practical output associated with work package 1 was to develop molecular diagnostic tools that could be used for the rapid analysis, identification, and quantification of different types of resistance in blackgrass. So for us, it's really important to understand what resistance mechanisms we're dealing with. So if you spray a field with a herbicide and you have weeds left after you've sprayed it, you can clearly see that this could arise from different mechanisms. It could be down to target site resistance. It could be down to non-target site resistance. In some cases, those two types of resistance often coexist together, so it can be down to both types working together. It's really important if you want to try and counteract those resistant weeds in understanding what you're dealing with. So you can actually make things worse if you don't understand the type of resistance you're dealing with. But establishing resistance mechanisms, as is currently done, is time-consuming and expensive. So very typically, an agronomist will come and collect seeds from the surviving weed plants at the end of the growing season, take them away and test them in their labs and glass houses by germinating them and then either spraying them with herbicides and seeing which herbicides they're resistant to, or in some cases, if they have the equipment to be able to carry out metabolism studies. And by combination of those different approaches, they can start to see whether they're dealing with target site resistance, non-target site resistance, or the two types working together. Importantly, and as you can probably see here, that type of information, not only is it time consuming, it's expensive. So these assays cost several £100s to perform, and the information is only available to you as a farmer for the following growing season. So if you're seeing blackgrass popping out the ground in the spring, you have no idea whether or not it's going to be resistant or not to your herbicide until you've actually gone out there and sprayed it, unless you have access to a molecular diagnostic. So what we wanted to do was kind of take this fundamental knowledge that we're starting to generate about understanding the molecular basis of herbicide resistance in blackgrass and develop a practical pocket diagnostic. And there are different ways of developing biomarkers for different types of resistance, some of which rely on analyzing the DNA, some of which rely on analyzing the proteins that are expressed in the plant, and you can also use the metabolites which accumulate in plants as well, sometimes to tell you which metabolic pathways are operating and which are not. So we took the kind of view that we wanted to use a combination of these different approaches to develop our molecular diagnostic tools. And in the case of the target site resistance, it was relatively straightforward because target site resistance, if you recall, is down to mutations in genes encoding proteins which are targeted by herbicides. If you can detect those mutations, you can, of course, detect the resistance, the target site resistance. The challenge for us in this instance wasn't the fact that this technology was difficult to do. We've known how to do these sort of sequence type analyses for many years. It was now to adapt technologies that were just being developed at that time to do this rapidly. So this isn't the sort of thing that your average farmer or agronomist is going to be able to do easily, but it is the sort of thing a reasonably trained scientist can do in just a little, about 40 to 45 minutes out in the field. And this is the use of isothermal amplification using what we call LAMP technology to generate, amplify up your sequences which contain your mutation and identify whether or not you've actually got your target site resistant present or not. And LAMP, of course, is one of those technologies that has also recently been developed for the application and detection of COVID-19 at the DNA level in people. So this is a technology that's kind of taken very much off over the last 2 to 3 years. And this is what it kind of looks like. You amplify up sequences. In this case, if you've got the your mutation, you'll amplify up a different product than you would do if you didn't have the mutation. And this can be seen from a colored marker that comes up on the LAMP apparatus. And it typically takes anything up to, as I say, 40 to 45 minutes. The beauty of the isothermal amplification is this is portable equipment. You don't need large, expensive PCR equipment, which requires mains supply. This stuff will all operate off a battery. What we were really interested in, however, was looking at non-target site resistance and a diagnostic for that. And we knew this was going to be a trickier thing to do because we knew that non-target site resistance isn't encoded by a single gene. It's going to involve multiple genes, multiple proteins. And so we had to do a whole search for different biomarkers, everything by looking from gene expression all the way through to the expression of different chemicals in terms of metabolites. We latched on at the end to looking at proteins. And the reason we did that is we started off by doing some mass sequencing studies. And we found that, in fact, up to 4,000 genes might be different between a resistant and a susceptible blackgrass plant. And we knew that a lot of those genes were being upregulated for reasons other than resistance, because all of these blackgrass plants are genetically very heterogeneous. They're very different from one another. So there are hundreds and hundreds of genes that would differ between just two normal blackgrass plants anyway. So it's very difficult to tell which of these genes were indeed involved in resistance and which of them were just involved in the natural variation between different individuals. However, when we got down to the level of protein expression and we started to tease out all the different proteins and separate them out utilizing electrophoresis, we could see that, in fact, a very small number of proteins were reliably upregulated in non-target site resistant plants. In fact, we got down to about 20 different proteins that were always associated one way or another with different types of non-target site resistance, but with only one protein which was always expressed in non-target site resistant populations, irrespective of the different phenotype that we saw. And so this protein, to us, was an ideal biomarker for detecting non-target site resistance. It was a protein that it is called AMGSTF1, so that's Allopucurus myosauroides glutathione transferase 5-class 1 protein. I'm not going to go into what that protein is. It's just this biomarker is critically linked and causatively linked to part of the non-target site resistance phenotype. And we know it is because we can take the gene that encodes AMGSTF1 out of blackgrass plants and we can put it into other species. And when it's expressed, we can now see that these other plants become resistant to multiple herbicides. So that tells us that it's an important component part of non-target site resistance in blackgrass, again confirming its value and importance as a biomarker of this type of resistance. So we were then interested in taking this knowledge. We know that AMGSTF1 is a molecular biomarker of non-target site resistance in plants. How can we turn that into a low-cost, rapid diagnostic? And the technology that we kind of adopted, and which is one which we're all now very familiar with, are lateral flow assays. So lateral flow assays are immunoassays. They use antibodies to detect the protein of interest. Of course, we utilize them all the time now for detecting COVID-19. But what we adapted the lateral flow device to do was to detect the presence of AMGSTF1 in blackgrass plants and then to be able to read off the amount of that protein to tell us whether or not that plant was resistant to multiple herbicides or not. How the lateral flow devices work, of course, is in the instance that we used two different antibodies raised against AMGSDF1. One of them we printed onto a membrane as our test line, and the other antibody we put in the sample application well joined to a dye. And the idea is that when you put your protein extract from your plant onto the lateral flow device, AMGSDF1 will move up the strip until it hits that printed line on the antibody where it gets bound. Then your second antibody comes along and binds to the other side of the protein with its dye, and effectively gives you a typical test line, again, which we're all very familiar with in terms of lateral flow devices. The assay itself is very straightforward. You just take a blackgrass plant, you snip off about 2 to 3 centimeters off the end of a leaf tip, put into a plastic pot with some extraction buffer and 2 steel balls. You shake it to smash the plant tissue up. You extract the plant tissue, spot it onto the lateral flow device, and then let it develop. And about 20 minutes later, you can then put an optical reader. This red device that we show here is actually an optical reader which is specifically for lateral flow devices. I'll come back to a kind of adaptation to that we then subsequently have made this simpler to use with respect to utilizing mobile phone technology. But effectively, this is the basis of a very simple lateral flow device which costs about £3 to £4 to manufacture per device, and which we have now sold, which we now market through our partners MoLogic as a commercial product. So I'm not doing the commercial sales here because, you know, believe me, I'm never going to get rich out of selling these particular diagnostics. But I think what it is, it's a nice illustration of doing some basic science that started off in 2014. And by 2017-18, we are actually commercialized and started to develop these MoLogic-based lateral flow devices, which are now available to UK farmers. So it's great having these lateral flow devices, but do they work? So the best way that we could demonstrate this was by working with our colleagues at Rothamsted, who harvested 190 different populations of blackgrass from across the UK, showing different levels of non-Targetsite resistance. And they showed that have absolutely excellent correlations and associations between the levels of expression of this protein as judged by the optical reader on the lateral flow device and the levels of resistance that we saw towards different herbicides. So the science, actually, the technology has been demonstrably kind of scientifically shown to be rigorous. So we have the lateral flow device. What can we use it for? So I'm just going to run through some things coming towards the end of this presentation, which just tell us how we can take a piece of technology like this and adapt it to make it more useful to a wider audience, and also to use it as a practical tool to counteract a major problem in agriculture. So first of all, we asked the question, could we use it in other wild grasses? Because blackgrass is a big problem in the UK. It's actually also a problem in Northern Europe as well, but it's not much of a problem in North America, and it certainly isn't a problem in Australia or other large grain growing areas around the world. The second thing that we asked about was could we adapt the lateral flow device to be used with a smartphone reader so that we didn't need the specialist optical reader that we'd originally developed it for. We then wanted to know what we could use it for in terms of practical resistance management out in the field. We also could then think about how we could use the core knowledge that we developed about resistance mechanisms to come up with new chemical technologies that would allow us to bust effectively or counteract herbicide resistance out in the field. And so it's not directly linked to the application of the diagnostic, but it is a spin out of the knowledge that we gained from understanding that AMGSDF1 is a very important protein involved in herbicide resistance. And then finally, could we use all of these diagnostic technologies that we're developing in integrated weed management? How can we actually practically give these tools to farmers in order for them to be able to use them out in the field to counteract herbicide resistance? So the first question was, could we use it in other grasses, wild grasses? And the answer to that is, although these wild grasses have in many cases evolved quite separately from one another, many of them do have a protein that closely resembles AMGSTF1, and in some cases so closely resembles it that we can use the antibodies that we generated for blackgrass as effectively proxies to recognize the same protein or the orthologous protein in other species like ryegrass and wild oats. So for both ryegrass and wild oats, we have been able to manipulate and change the application of our lateral flow technology so that we can also use the same LFA approach to detect non-Targetsite resistance in other wild grasses. And that's important because if you're a farmer in Australia, you're probably more interested in controlling ryegrass than you are in any other weed. And likewise, we've been working in North America and controlling wild oats in barley. So, as I say, we've adapted the technology now to work in wild oats, Italian ryegrass, rigid ryegrass, and we're working with agronomy companies in Australia, Canada, Spain, and Israel. So it's not just a UK-centric project now, it's one that's spread all over the world. The second thing that we kind of asked was, can we use the technology in resistance management, and how can we understand the effects it has on different types of management strategies as to how we're driving up or down non-Targetsite resistance? So very typically, one of the ways in which you can try and counteract herbicide resistance in grasses is by rotating modes of action. In fact, you can use different modes of action within the same growing season in rapid succession, one after the other. So you're changing the way in which you're trying to hit the weed and keep it from developing Targetsite resistance. Well, it works at one level. It may prevent not Targetsite resistance from developing, but what we discovered was it also drives up the rate at which you see non-Targetsite resistance developing. So we challenged the perception. In fact, this is the best way of dealing with herbicide resistance because it has a knock-on effect of driving up the rates of non-Targetsite resistance. And in many ways, non-Targetsite resistance is a longer-term, more difficult problem to deal with than Targetsite resistance. So, it's one of the things that we've advised farmers to move away from as a management strategy. And the other thing we've been interested in is just seeing how long-lived the non-Targetsite resistance phenotype and genotype is. So if you remove herbicide selection pressure from blackgrass by no longer spraying against it, can you reduce the levels of non-Targetsite resistance by reducing the expression levels of AMGSTF1? Unfortunately, at the moment, the answer to that appears to be no. We've done this over several cycles now and have not seen the level of the protein decline with the removal of the selection pressure, which suggests to us that, in fact, the AMGSTF1 protein expression does not bear a fitness cost to the grass weed. We have been more successful in adapting the technology for application utilizing smartphones. So what we've done here, we've actually changed the format that the lateral flow device works in. So you take the lateral flow device effectively out of the plastic housing it was in originally. You put it on what we call a dipstick format. And in the dipstick format, you stand the dipstick in a solution of your blackgrass extract. You allow then the strip to develop exactly the same way as you would do for a normal lateral flow device encased in a plastic shroud. But in this instance, you get you can take an image using a smartphone camera, which you can then analyze on an app available through MoLogic, which then gives you exactly the same kind of information that you would be getting formally from the optical reader, which you would need to physically buy in order to be able to do your assays. So this is a way of kind of making the technology more accessible and also reducing the overall costs down, both of producing the lateral flow device and indeed the way in which you analyze for resistance. Now, in terms of where we go with the in the future, one of the things that we were able to do with the protein AMGSTF1 was work with structural biologists to solve its three-dimensional crystal structure. And when we did that, we found some interesting binding sites on the outside of the protein, which we're then able to chemically modify utilizing chemicals that are currently utilized as experimental drug molecules. And one of these was actually able to covalently modify the outside of AMGSTF1. And in doing so, we found that when plants were co-sprayed with this and the herbicide, we could reverse the resistance phenotype. So what we've got here is what we call a herbicide resistance busting chemical, which you can help restore chemical control by applying this alongside your herbicide, and then the herbicide will work again as it would do normally. Now, the interesting thing about this and comes back to the fundamental science is something very similar to this happens in human beings when we get resistant to anti-cancer drugs when we're being treated with drugs to prevent cell division. Our cells start to produce a protective protein, which oddly enough is another glutathione transferase type protein, which is evolved totally separately from the blackgrass protein, but which it does something really very similar. It counteracts the effects of the chemical largely through detoxification, and then we can also inhibit it in the same way by utilizing these chemical modifying drugs. So effectively, it's this concept here of resistance busting as being a new strategy for reversing non-Targetsite resistance towards herbicides in much the same way as has been adopted in medicine to counter out multiple drug resistance effects of AMGST, the glutathione transferase present in cancer cells. So finally, what we've been interested in doing is trying to pull together all of these different diagnostic platforms into one workable tool. So we've got a DNA-based diagnostic that tells us about Targetsite resistance, a protein-based diagnostic that tells us about non-Targetsite resistance. So we could bring those two things together with a lot of clever kind of modeling into understanding what the overall levels of resistance would be towards different herbicides. And the long-term goal here is that we would then roll all of this stuff into effectively a management platform, which utilizing app-based technology would allow farmers to make detailed decisions based on the readings that they were getting, which would then enable them to come up with an integrated weed management strategy, i.e. which is the best chemical to treat these particular weeds, or indeed which is the best management strategy they would want to adopt, which might not be using chemicals at all. So it's what we call the digital agronomist, which is a piece of work we're currently kind of developing with the app developers at the moment. So that brings me to the end of the presentation. I'd like to thank the team involved in helping us develop both the molecular diagnostic and do the underpinning science. In particular, I'd like to thank Naopo Nkokusung, who's been particularly instrumental in helping us develop those applications for the LFA technology overseas as well as here in the UK, and for the AHDB and BBSRC support for the work, which enabled us to do all the kind of core basic science to get things underway. So I think that's it from me, Derek.
Many thanks, Rob. I think that was both a scare story and a bit of a reassurance at the end there, to be honest. I've got lots of questions to ask, but I thought maybe I'll leave it open to others. Has anyone else got questions they'd like to ask? Rob?
Should I stop sharing it? That's right.
Yeah, you might as well just now. So Rob Hancock.
Yeah, thanks for the presentation, Rob. So I was interested in this this non-Targetsite resistance, and you said originally before we started evolving weeds that were resistant to herbicides, originally the reason that these herbicides were selected was because in crop plants the detoxification pathways were upregulated relative to the weeds.
Yeah.
So I wondered, is that something that's been inadvertently selected during domestication? Why do we see this difference between crop plants and similar weed plants?
It's $1 million question actually, and you're quite right. Yeah, I'm just trying to work out how to stop sharing actually.
Oh, just leave it. You're fine.
Okay. Yeah. So basically, we don't understand why those enzymes are there. We've actually done some work with some ancient crop varieties, and we've discovered that it's got nothing to do with anything that was done in terms of domestication of crops after the arrival of herbicides. So it's something you can go way, way back and find, the original wheat lines that were domesticated in the Victorian times. They have higher levels of these enzymes as well. I think it's got something to do, the enhanced detoxification must have something to do with enhanced stress tolerance, or at least maintaining yield under difficult conditions. But we do not understand the basis of why that has already been pre-selected. It is $1 million question, and you see it in all the major crops. It's not just wheat. You see it in maize, and you also see it in soybean, cotton, the whole lot.
Okay, so is that constitutively expressed then? Or
Yes.
It's constitutive.
Constitutively expressed. So there doesn't seem to be any, there doesn't seem to be a penalty associated with it, and it's a whole gambit of enzymes. It's not just one or two. It's just the whole system is upregulated.
Okay, thank you.
So, any more questions? I'm trying to see if anyone's got a hand up. I don't think there is, so that allows me to maybe ask some. Do you think there's a problem as well there with? I mean, he was talking about the crops are really not delivering what they used to deliver. Presumably, there's also the problem that actually altering crops takes a long time, and the weed problem seems to evolve even faster than science genetics and the crop side can do it. So we need all the plates spinning at the same time.
Yeah, I mean, the other thing is, about herbicides, and it's not something we can use GM technology. Well, you can use Roundup Ready technology, I suppose, which has been mooted as a means of controlling herbicide resistance. But even then, glyphosate resistance in weeds can evolve very quickly. So it's difficult to see how you can apply genetic technologies in crops to kind of help out on the weed control side.
So is there anything on nature-based? So you could have nature-based solutions that would selectively go for the weeds rather than the main crops. But again, I suppose the distinction between them is very fine.
There's been interest in the past on developing things like mycoherbicides, where you use herbicides which are basically fungal based, and which are selective for specific types of weeds. But most of them they've proven to be useful in horticultural applications, but are prohibitively expensive, or they don't really work as fast as they need to work when you use them in arable settings.
Mm-hmm. Okay. Maybe I can ask one of the more detailed ones. The stuff you did in Arabidopsis looks really interesting. So what do you think's happening with the upregulation in anthocyanins and flavonoids now?
Yeah, I didn't really touch on that because I suppose it's a different. But it seems to be something that the resistance mechanism is underpinned by an enhancement in antioxidants. And so what we see is an enhancement in thiol antioxidants, an enhancement in specific types of flavonoids. And it sort of starts to beg the question of whether or not those increases in flavonoids are in any way involved in enhancing herbicide tolerance. Because some of the enzymes that we also see induced in the early stages of resistance development are linked to flavonoid metabolism.
Because it sounds, I mean, I've done a lot of work a long time ago on dietary routes to controlling cancers, and certainly when you put flavonoids in there, what they do is they G up the human's innate anti-cancer and detoxification systems because they're sacrificial antioxidants being generated. So you're just wondering if the production of antioxidants are helping the innate defense mechanisms of the weed.
I think that's exactly what is happening, Derek. I mean, that's kind of what we're working on a bit of that at the moment, and trying to work out effectively. Well, I think if you think about that, resistance here is being delivered by multiple mechanisms within the cell, and I think this is one of them.
So if I can exploit this, and I'll keep asking questions for a wee while until people. Oh, sorry, there's one from Richard Pickering. Richard. Are you there, Richard? You're on mute just now. Still there, Richard? Still on mute. It doesn't look like you're on mute anymore, Richard, but I don't think we can hear you. If we're struggling, stick your question in the chat and I can ask Rob. What was the other one I was going to ask? Yeah, so I suppose certainly interestingly, if you're talking about guided drug design or treatment design, one of the spin-out companies from the local university, Dundee University, is Accenture, and there's very much an AI-guided drug design. Presumably the big money must be going into that now for agrochem.
it will be, but for mode of action, I would work, I think.
Yeah.
I mean, interestingly, I think it's only recent, the penny's only really relatively recently dropped, that if you don't deal with this resistance mechanism, it's actually going to take your compounds down before they've even been developed. Because most herbicides are, most selective herbicides will have an element of detoxification involved in how the way they work between different plants. So if you've got a metabolism-based resistance mechanism and it's agnostic of the different chemistry it works on, you could end up with a chemical that you spent hundreds of millions of pounds developing. You spray it onto a weed that's never seen it before, but it's already resistant to it. In fact, they've seen a little bit of that in Australia, actually. So it's a real big problem.
Yeah.
But yeah, I think we would be really interested in going down that route to discover much more benign chemistries that reverse resistance, because the problem with the chemicals that we've been using is you wouldn't be allowed to spray them out the field. They're just they're basically alkylating agents. You know, they're too toxic.
Yeah, it's sledgehammer effect, isn't it?
Yeah.
So we've got the question for Richard. He's saying, how long is the viability of blackgrass seed in the soil? And could a change in rotation reduce the seed bank?
I don't know what the latest. It's several years. I think it's at least 7 or 8 years. It depends on the conditions. But that what farmers are now doing actually in really badly affected areas is they're stopping growing cereals and they're putting it back to pasture, and then they'll put it through several years worth of cattle or sheep grazing on it to reduce down the seed bank. But the problem with that is it's great if you're on a mixed farm, but actually for a lot of arable farmers in East Anglia, that's not what they're set up to do. So it has
like the equivalent of potato cyst nematode. Once you've got it in, it's almost impossible to get it out, or you're 10 years before you can do anything again.
But there's been some quite interesting work done on where they try and flush the blackgrass seeds into germinating, utilizing some chemical treatments, and then you can hit it with glyphosate when it all starts to germinate.
Yeah.
So you can just use it as a kind of flushing mechanism.
So I think there's another question from Rob Hancock. He's asking what genetic or genomic resources are available for weed species, and how could they be used to develop new herbicides? Is there much out there?
They've recently sequenced the genome of blackgrass. I think both Bayer and I think BASF have both published it. I suspect they'll have the other genomic sequences for the other major problem weeds like Palmer amaranth and maybe for Lolium as well. To my knowledge, I don't know how they're using that information in terms of new product development.
So do you know if they've got sort of multiple sequences? Because I mean, that's obviously where the interest starts to lie, isn't it? If you can start to compare resistant and non-resistant species, accessions, etc.
Yeah, I'm not familiar with that, Rob. But the only information that we've generated has all been transcriptome data.
Right. Okay. All right. Thanks.
Okay, well, I think we've questioned you enough, Rob. So thanks very much for a hugely informative talk in an area that I wasn't that aware of, but I'm hugely informed now. So if everyone could show your appreciation the way we normally do, a bit of hand clapping wouldn't go amiss. So thanks very much, Rob.
Yeah, thank you very much. Thanks, Derek. Thank you all.
I'm sure we'll see you again.
Yeah, you will.
Just to finish, we will be having another APGC talk on, let me have a look now, 14th of April. And it's Mark Tester from the King Saudi University. He's talking about sustainable controlled environment agriculture in the extreme climates. Rob set a high bar, so let's hope Mark can hit that. So thanks to you all. Hopefully we'll see you soon.
Thank you, Derek. Thanks all.
Right, thanks, Rob.
Yeah, bye.